You got to work it. Okay, awesome. All right, so we're going to go ahead and get started. As always, we got a good one for you today. What kind of a snake do you find on a car? A windshield viper. Good, good. I don't like snakes. Don't like snakes. Can't say that enough. All right. So one thing we're going to have to change is, so with iClicker for attendance, you can either be virtual or you can be in the classroom and still click in, right? So if you're on quarantine or whatever it is, you still get credit for being present, which I like, right? You like. But for contact tracing purposes for the university, I have to keep a record of where everyone is sitting every day. So our new normal is going to be when you come in to go to the seating chart, and the seating chart will be blank and you put your name in. That way, if somebody gets marked as present on iClicker, but they're virtual because they're in quarantine or whatever it is, I know the difference between you were here and participating because you were on iClicker, or you were here in the classroom and exactly where you were. So I'm sorry, but we're going to have to do this every day. So go to the seating chart. Now I'm going to give you a little hint here. I think a few people had a problem in the beginning when you were entering your names on the seating chart because you didn't hit enter or click on a cell, a white cell that wasn't the cell that you were typing in. So it didn't save it. So what I want you to do is to type in your name, either hit enter or click out of that cell, close it, then go back to the seating chart and make sure your name is there. because it's super, super, super important. Okay. So every day, I'm sorry, we will do this. It happens. So give everybody a chance to do that. Yes. Yes, because I want to give people who are out the chance to still be participating and to get credit for being here. So I'm kind of stuck that I have to kind of make you do both. I'm sorry. Maybe other teachers have better solutions than mine, but this is what I could come up with. So it is what it is. So please take a second and make sure to close and then go back to the seating chart and make sure it's there, because I'm seeing some empty names where I'm seeing people. So we need to make sure that we have the right things done. So please make sure, so it should be all the way full in the front, and we shouldn't see empty chairs until towards the back. And I'm giving y'all a minute to do it. Hopefully we'll get quicker. I'll have it up when you come in. So as soon as you come in and sit down, go ahead and put it in. Okay, anybody having issues? They need help? Everybody good? Okay, well, I guess while we're waiting, I'll give you another joke. What do you call a sleeping dinosaur? A dinosaur. It's quality, right? It's quality. No, it's really quantity over quality. But that's okay. All right, so we good? Please make sure your name's on here. It's really, really important. Really important. Okay, so keep doing it. If you're still working on it, keep doing it. All right, and then we are going to pick up kind of where we left off, and don't forget to check in on iClicker as well. Yes, ma'am. What reading was this open for each chapter? So one chapter, when one chapter ends, the next one opens. So we got to finish one, and then two will open. So they go one at a time. Okay. Full screen. Here we go. All right, so remember last time, this is the last thing we ended on, right? And it was signal transduction. This was what was really important for communication between cells of the same organism. So what were the two results like of a ligand binding to a receptor? What were the two ways that we can induce a signal transduction now inside the cell? So binding does what? That's right. So you get enzymatic activity of the receptor. Good. Okay. That's one. Protein association. R-O-T-E-I, protein. And so all of this is intracellular, right? On the inside, because your ligand was where? Outside, and you need to now propagate a signal inside. So all of those changes, right, have to be propagated across the membrane. How do you do that? What's a characteristic of a receptor? It must what? Span the membrane. So it must have an extracellular region and an intracellular region, right? For that to happen. Okay. The next slides in this section will go real fast because they're easy. But I still see people copying. So once we have all of our specialized cells, because we said those were the two things that were really important, having specialized cells and then having communication amongst the different cells of the organism. So then we can organize those cells into tissues, we can organize those tissues into organs, we can organize those organs into an organism, right? So your organism must have then specialized cell types, just like we said, kidney cells, liver cells, whatever cells. And so this will allow our multicellular organism to respond to a plethora of environmental stimuli, right? That's the name of the game. Because what happens if we reach homeostasis? If you reach equilibrium with the environment, let's say I reached equilibrium with the environment, is that a good thing or a bad thing? Really? Should my temperature be the same thing as my environment? No. Should my oxygen content be the same as the environment? No. So I know that when we say homeostasis, right, you think it's a good thing, but if you think about it in terms of homeostasis with the environment, that means you're dead. It's true. We just went downhill real quick, right? But that's the point. We want to maintain homeostasis inside of our bodies, yes, but not with the environment. We need to maintain certain things inside of the organism in order to survive. If we have homeostasis with the environment, we're done. We're cooked, right? So we have to be able to adapt to changes, right? If it gets cold, what do you start doing? Shivering, right? Because you don't want to be at equilibrium with the cold. You want to be warm. So you're going to shiver in order to produce heat, right? So we're going to adapt to those changes through signal transduction mechanisms. That's how we're going to signal our body to, hey, those muscles need to start contracting so that we can really start shivering and warm up, right? Not really a August topic, but you know. So some other examples, right? If we look, one of the big things that we have to worry about as an organism is getting all kinds of macromolecules, getting nutrients, getting oxygen to every part of the body, right? So our circulatory system, which is both our, what? Heart and lungs, so that's part of it, that's true. So you circulate things through the blood, so that's cardiovascular. And what else? Lymphatic. You also have a lymphatic system. What cell types typically are in the lymphatic system, right? You have lymph nodes. So if you get sick, all your lymph nodes will swell, right? You know this? So what kind of cells are typically distributed through the lymphatic system? Immune cells, right? Various immune cells. So circulatory kind of encompasses both of those. So if you look at just the circulatory system, right? What are we going to be transporting? We've got to transport things that we consume to all the different parts of our body, right? So they can have energy. We have to transport oxygen. If you don't have oxygen, you're not doing what? You're not doing cellular respiration, right? Breathing is getting the oxygen into your bloodstream, but then once that oxygen gets dropped off wherever it's destined to go, then it's going to be used for cellular respiration. Hormones. Hormones are super important because a lot of times they're produced in one place in the body. Maybe they're produced in the brain, but then they need to work on a different organ. So they have to be transported all the way across the body, right? So maybe we want to stimulate the digestion process. So that hormone is going to go to the intestines, right? So you just got to kind of think about, it's all interconnected, but y'all already know that. Y'all are so smart. And then a bunch of organisms come together and we make an ecosystem, right? So this is our highest level. This is our biggest thing. We have multiple organisms living together in the same environmental niche. We have a nice environmental niche here in the classroom. So we have a shared use of consumables, of resources, and of waste management. So what happens when all that goes awry, right? Right, know what that is? Those are algal blooms, right? This is bad. Why is this bad? Because you end up with a lot of dead fish. Why does an algal bloom result in dead fish? Actually, not the algae that deplete the oxygen. The bacteria that eat the algae deplete the oxygen. And then the fish have nothing to breathe, right? So it gets really, really gross, really pretty quickly, right? It's really pretty disgusting. Okay, so on to some of my favorite stuff. Genetic information. Love genetic information. So in the 1940s, 1950s, they were really trying to figure out what is the source of genetic information. And we really didn't think it was DNA to begin with. Why? You have to store a lot of different genetic information, right? We know that we have thousands of genes. That's a lot of information. But how many individual component molecules make up DNA? How many different nucleotides do we have? A, T, C, and G. So how many you got? 4, okay? A better candidate in scientists' minds in the 1940s, 1950s, and before that was that it was proteins. Why do you think that was? You got 20 building blocks, right? So you got more diversity, so you could store more information. But it's not true. It actually ended up being DNA. And so in the 1940s, 1950s, they were trying to figure out what's the storage of genetic information. They sort of figured out it was DNA, and then they said, okay, what's the structure of DNA? So, and everybody knows the famous story of Watson and Crick, right? But I got a problem with the Watson and Crick story, right? Have y'all heard this? Yes, from a couple of different people? Good, good, good. So it was really Rosalind Franklin who collected the data that led to the structure of DNA. And she actually collected X-ray diffraction. And this is her beautiful photo 51 that she collected, that you can measure the distance between the helices. And so we know that this is 34 angstroms. And that distance repeats itself over and over and over again. And so from that, basically, they were able to figure out that it was a helix. So this is 34 angstroms basically per turn, per twist of the DNA. So Watson and Crick get the credit for it, right? They got the Nobel Prize for it. But really, she actually started publishing on the structure of DNA before Watson and Crick did. And she collected the data and she analyzed the data, and Watson and Crick and she separately published in Nature in the same volume. So it came out the same day. They put Rosalind Franklin's paper behind Watson and Crick's paper so that it was then showing it supported Watson and Crick's conclusions. But really, it was Rosalind Franklin who collected the data, analyzed the data, and came to the conclusions. Really, Watson and Crick, it's debatable whether they stole her data or not, but regardless, it was not handled well. Put it that way. It was really not handled well. And they looked at her data and some of her conclusions and then based their model on that. So she really got the short end of the stick, because when came time for Nobel Prize in 62, Watson and Crick were awarded the Nobel Prize. And it was really Watson and Crick and Watkins who was working with Rosalind Franklin. But you know, she had gotten cancer and died before the Nobel Prize. Now you'll read, right, that they never award a Nobel Prize I always say it wrong. Yes, thank you. when you're dead, they won't give you the Nobel Prize. But honestly, that rule didn't take effect until 1970 something. So that rule wasn't even in effect when the Nobel Prizes were issued. But there was a rule then that you couldn't have more than 3 people get the Nobel Prize. So they left her off. But anyway, okay, so DNA is a double helix. That's our big takeaway, right? And so we know we've already covered what's a nucleotide, right? And we know the difference between a deoxyribonucleotide and a ribonucleotide, right? Which carbon? Carbon 2 of the ribose, right? So the 2' carbon is a hydroxyl in RNA and a hydrogen in DNA. So everything else is structurally the same except for that. And we'll talk about why that chemically, why that's really, really important in the next chapter. So we know our complementary base pairs, right? CG and ANT, cars in the garage and apples in the tree. You heard that before? Yeah, some people. So cars in the garage and apples in the tree. That tells you your pairing rules, right? And then you have to know the purines and the pyrimidines. How do we know purines versus pyrimidines? What is cytosine? Cytosine is a one ring is a what? Pyrimidine, Pyrimidine, pyrimidine. Those with 2 are what? Purines. P-U-R-I-N-E-S. P-U-R. Come on, won't even let me erase. P-U-R. Purines. So it's a little bit counterintuitive, right? The longer name, you only have 1 ring. The shorter name, you have 2 rings, right? So keep that kind of in mind. So how's it different in RNA? There's no thymine. What do we have instead? Uracil. How is uracil different than thymine? If I wanted to turn this thymine right here into uracil, what do? Take away the methyl group. Now I have uracil. Okay. So how do we designate the difference between these base pairs? What do you look for? Right, we say sometimes DNA is GC rich or it's AT rich. We're counting hydrogen bonds. We're counting hydrogen bonds between the base pairs. So GC is how many hydrogen bonds? 3, and AT is 2. What holds DNA strands together? Base stacking. Lots of people want to say hydrogen bonds, right? But what kind of an environment is DNA in? In an aqueous polar environment, right? So basically there are water molecules everywhere. So making a hydrogen bond with another base is cool. Making a hydrogen bond with water is cool. The DNA doesn't really care, right? It doesn't prefer a hydrogen bond with one substrate or another. But when the DNA is together in that 3-dimensional shape, the nuclei of the atoms of those nucleotides are close together. They're in that perfect sweet spot. Y'all know about van der Waals forces? You learned about that in Chem 106? Yeah, okay. So, right, you know that if they're far apart, they're not attracted to each other. If they're too close together, they repel each other. But there's this sweet spot where they're perfectly attracted to one another, right? That's base stacking forces. Okay, so that's what really holds together DNA. I'm giving away all the key things at the beginning. All right, so you've seen this probably picture of DNA how many times in your life, right? Now you know about the numbering of this ribose ring, right? So why Do I say that this strand has polarity from 5' to 3'? The polarity is telling me basically which end is exposed. So we start with something attached to what? If I count my carbons, that's 1, 2, 3, 4, 5, and these are primes, right? So here my phosphate is attached to the 5'. They put the 5' here, but what does the 5' really refer to? It's the carbon that's sticking out of the ring that's really part of the ribose, right? So that's where the 5' comes from. It's so disturbing to me that they put it here because then it makes you think it's associated with the phosphate group, right? Yeah, but it's not. It's associated with the ribose. Okay, so now why do we call this the 3' end? What's exposed? The hydroxyl group on the 3' carbon. That's why we call it 5' to 3'. So one strand has one polarity and our other strand has opposing polarity, right? So they are called anti-parallel. The anti-parallel refers to that polarity. And so that's why, because they're perfect mirror images of one another, that's why we can have DNA replication, because each strand can make a whole new strand, right? So we have that semi-conservative replication, which y'all did in biology, right? Yeah, okay, good. All right, so everybody's pretty familiar with central dogma, right? So it tells us how information goes from being stored inside of the nucleus to having a product inside the cell. So you can always start from DNA and you can make more DNA, and we call that replication. Or you can go from DNA and you can make some kind of RNA, right? The more traditional way that we think about is going to mRNA to then go to protein. So that's transcription, but that's not the only kind of RNA that we make. We make lots of different kinds of RNA, especially for protein synthesis, right? rRNA, we make a ton of that. tRNA, we're going to need for protein synthesis. Anybody know what microRNA? Yes? MicroRNA can actually bind to mRNA, and it can basically make it degrade and silence it so that it never gets turned into protein. So it's a way that we can regulate. Same thing with small nuclear RNA. These are different ways that we can regulate how much mRNA actually gets turned into protein. So it's another level of regulation within the organism, within the chromosome, whatever you want to say. So then when we go from our mRNA to protein, that's translation, and we can make so many different kinds of proteins. Why is it that we can make hundreds of thousands of different proteins inside the cell? What did y'all tell me earlier today? How many different kinds of building blocks are there for proteins? 20. Do you have any idea the variation that can occur when you have 20 different building blocks? And then you can have a protein that's hundreds, thousands of amino acids long. The variation is astronomical, right? And so because we can have all that variation, we can have proteins that have so many different functions, right? So this is your other biology review, right? We know that in the DNA, in the 2 strands, we have a coding strand, we have a template strand. The template strand will bind to what? To the mRNA. The mRNA will then code for the amino acid. So if you want to know what your mRNA strand is going to look like, you look at the coding strand, right? Yes? No? Seen this before, right? I love the head nodding or the shaking. Either one gives me information, right? So then we'll look at our amino acid sequence. So a lot of times in biochemistry, we not only look at how genes might be different from organism to organism, but sometimes you can have changes in the DNA that don't lead to a whole lot of changes in the protein. So sometimes it's important to look at the DNA sequence. Sometimes it's really important to look at the protein sequence because that can give you a lot more information. So recently, biochemistry has bloomed. I mean, the different divisions of biochemistry have grown a lot over the last probably 50 years. So we know that we can look at the genome, we can look at the collection of genes, but that doesn't tell us everything because basically every single cell in your entire body has the same set of genes, right? It has the same genome. That's the whole point. So basically every cell in your body has the same recipe book. But does every cell in the body cook all the same recipes? No, right? So we can look at this transcriptome of each individual cell, and we can look at which genes actually get transcribed, which ones are actually turned into RNA products. But then there's another level, right? Because I just said that those mRNA levels can be regulated. So then we can look at the proteome. So we can look at actually which proteins are produced, right? Because sometimes you're going to make an RNA product, and from that RNA product, you can splice it differently and you can make 5 different proteins. So the proteome gives you even more information about what's actively going on in the cell. So it's lots and lots of information going on. So remember I said that this complementation of structure and function drives almost all of biochemistry, right? So we know that structure determines function. So if you have DNA and that DNA codes for a protein that has similar structure, typically you're going to have a protein or 2 proteins that have similar function, right? So if I have a bacterial gene that codes for a ribonucleotide reductase, this is an enzyme that is important in the synthesis of DNA, of those DNA precursor molecules. And I have that same gene in mice. Usually it's pretty similar, right? Maybe 90% homologous. Well, that's enough homology that structurally, how do the 2 proteins look? Pretty similar, right? They have pretty similar structures. So they're going to have pretty similar functions. But there are exceptions to every rule, right? Sometimes you can have really similar structures, but have completely different functions. So here's an example of that. We have a porin channel. So this is a channel that would span the membrane and allow things to transition from, say, extracellular to intracellular space, right? So it looks like a lot of beta sheets that are lined up, right? That allow for a big open space in the middle and allow for those molecules to go through. Well, this looks really similar, right? It has those same beta sheets. It has some variable regions on the ends, just like this does, but this is not a pore. This is the green fluorescent protein. So, you know, if you can engineer, this is a gene that was found in jellyfish. And you can actually take that gene and you can put it into another organism's genome, and they will fluoresce under the correct lighting conditions. So like you can, they do this in the lab, but you can have mice that they're GFP mice and they fluoresce, right? And so you can do this as an indicator of protein function. So you can actually insert this gene at different places in the genome, and I want to know if this is being expressed, right? So is this gene being, where is this gene being expressed? Well, if I put it into the mouse as an egg, right, and I let it grow into an embryo, well, what if only the liver fluoresces? Well, then I know that's a liver-specific protein. It's not just cool, it's really informative too, but it is cool. It is really cool. Okay, so proteins have this insanely diverse set of functions that they can do, right? I mean, like really diverse. So how did we get this diversity of hundreds of thousands of different proteins inside the cell. And the answer, for the most part, is genetic drift, right? That DNA inside of our nucleus is not perfectly replicated every single time. We like to think it is, but it's not. Our replication machinery is not perfect. It has errors all the time, all the time. That's why we get cancer, right? You know, there are proofreading mechanisms that go in and are supposed to fix errors, and most of the time they do, but sometimes they don't. And sometimes it's bad, and sometimes it's really good. And so if you look at an example, right, here's a general wild type gene for an enzyme, any given enzyme, right? And so if we look at this second codon, that G right there, if we mutate it, if we have a single nucleotide change, one nucleotide can change what? Well, it could change the reading frame. In this case, it didn't change the reading frame. In this case, we're still in the same reading frame because everything before is the same and everything after is the same. So in this case, it's a single point mutation that is a missense mutation. Y'all have heard that, right? Okay. So we change one amino acid, right? And sometimes one amino acid is not a big deal. But a lot of times in an enzyme, there are particular residues in the enzyme that participate in the actual enzymatic reaction that happens, right? That's the pocket where the action happens inside of an enzyme. And in this case, right, we have glutamate. Glutamate has a charge of what? -1. You don't have to know that yet. It's on the screen. But it has a charge of -1. So a lot of times inside the active site of an enzyme, if you have something with a negative charge, then it's going to associate with a positively charged substrate, right? So it's going to help to facilitate that interaction. Well, if I mutate that glutamate to a lysine, what's my problem? I now have what kind of a charge inside the active site? A positive. Well, if my substrate is positive and my active site is positive, are they going to associate? No, opposites attract, same repels, right? So now it's no longer going to bind the substrate. That's a big problem, right? So sometimes mutations are silent and sometimes they are not, right? And then they result in a defective enzyme. So when we talk about mutations, some mutations are ones that can get passed down and then create changes in species, and some will basically die with the organism, right? So mutations can be either germline or they can be somatic. So germline, when we talk about germline, this has to be passed from parent to offspring. So what cell types are we talking about? Gametes, eggs and sperm. Those are our germline cells. So if we have mutations in those, in that DNA, then that change is going to get passed on from parent to offspring. Every other mutation in every other cell is going to be a somatic mutation. So it can't be inherited. It's limited to this organism, right? So for example, my birthmark is an example of a somatic mutation. So I had a mutation that occurred in fetal development, right, that basically turned a protein to a permanent on switch to make blood vessels. So that portion of my skin has the gene and it's stuck in the on position. So I periodically actually still have to go and get laser treatments to basically kill the cells and break down the cells so I don't get, they call them blebs. So the skin will actually continue to grow and you'll get kind of like bumps and stuff because it's the blood vessel's still growing. They're still stuck in the on position. But I can't pass that mutation on to my son. It's stuck with me, right? Because it's in the skin. It's not in eggs or sperm, right? It's different. So how many of y'all heard about CRISPR? like totally cool, right? CRISPR is totally cool. It's a way, it's a mechanism that we can go in and we can target a particular piece of DNA. We can cut it out and we can put in what we want, right? CRISPR is amazing. CRISPR, I really think is going to be a big wave of the future and allow us to do things in medicine that we've never been able to do before. And it's already been used for that stuff, right? But here's the problem with CRISPR. You got to really think about it. You guys are the voting public, so you really need to understand what's at stake and how we regulate basically science bioethics, right? How do we know that changes that we make are not going to alter the future of an organism detrimentally, right? If I say, okay, I know that somebody's going to have, I don't know, Parkinson's, right? And as an embryo, I can go in and I can fix that. Absolutely, right? Can we please save somebody from that fate? Let's do it. But what if we make a change trying to prevent something that's not necessarily bad, like a birthmark, right? Or we can just go into the DNA when Dr. B was an embryo and fix it, so she'll never have a birthmark. But what if that changes something else that we weren't prepared for, that we didn't know was going to happen? And if that change occurs on the embryonic stage, it can be in the germline. So then I would pass that change on to my offspring, right? Right? Now we're getting in kind of murky waters here, right? And then what's a disease that needs to be fixed and what's cosmetic, right? What should insurances cover and not cover? And you'll see that CRISPR is amazing, but it's opening up a Pandora's box of bioethics that we don't have the answers to right now, right? And so I kind of think that we can use germline and somatic cells as a kind of division, right? If you just change somatic cells, is that change going to be passed on to offspring? No. So do whatever you want, right? But if you change the germline, you're actually changing human evolution. I mean, like this is huge. So anyway, that was my offshoot for the day. Sometimes, you know, you just think about these things and I can't help but share them with you. Right? It's really cool. Don't we want to help people, save people? It's been used to treat sickle cell anemia already. I mean, the potential is amazing, but we have to learn how to use things ethically and morally, and that's a societal thing that we kind of have to figure out. Okay. Really went off on a tangent there. All right, so we were talking about mutations, right? We were talking about mutations. So if you look at the DNA, mutations don't happen at the same rate across the entire genome. There are places where mutations occur at a faster rate, and there are places that mutations occur at a slower rate. So the places where you see a slower rate of mutation, those tend to be coding regions of DNA that make protein, right? Because if you change those sequences, there's a possibility that those changes will be detrimental and will therefore lead to the organism not surviving, right? So you need to make sure that if you have a mutation, it's not one that's going to kill the organism. Because if it does, that mutation won't propagate. But what about the sequences that flank a gene? Sometimes the sequence doesn't matter, it's just the distance that matters, right? You need a particular spacing. That can be really important in DNA. And so if the spacing is important and the sequence is not, guess what you're going to end up with? Lots of mutations occurring because it doesn't matter. So it's all about natural selection. You have random mutations that occur all over, but we only see the majority of the mutations in places where the sequence isn't that important, right? So if you look, the red is like a gene, right? Maybe it's a gene that we find in bacteria. And through lots of time passing, right, get to eukaryotic humans, we have genes that look gosh darn similar to a bacterial gene. Why is that? Because in the red, right, this is the coding sequence for that gene. If it changes, we can't make DNA anymore. Well, that's not going to survive, right? If you mess that up, it's not going to survive. The regulatory sequence that binds transcription factors, things like that, upstream and downstream. The light red is going to be really important too. But a lot of times, upstream and downstream, it's just spacing. And so you'll see lots of mutations in those flanking regions. So another thing that happens that's not necessarily a deletion, but can lead to a lot of variation in species is when we have gene duplication events. So sometimes when copying the DNA, the mechanisms that do the copying insert an extra copy of a gene. So it's a complete duplication. And there are different things that can occur after that duplication event. So sometimes if you have two genes, now I can make twice the amount of protein. And that protein is really beneficial and it's really great. And so we're going to keep both genes just as they are, and that's going to help this organism survive better, right? You can have another thing where you really only need one. And so that duplicated gene is allowed to mutate and to change. And so you actually end up with a new gene because it's making a different protein, right? So accumulation of mutations occurs, it changes, and that's how we can get new genes appearing. A lot of times it comes from gene duplication events. And then sometimes it's not more beneficial to have that duplicated gene and it gets deleted. So there are a lot of different things that can happen. So when we talk about genes, like when we said, okay, do I have a gene that looks like a bacterial gene, right? If we say yes, our ribonucleotide reductase, right? What do we classify that as? Like me and bacteria have similar genes. We call those orthologs or orthologous. Those genes are orthologous or they are orthologs. Woo! So you can have orthologs and you can have paralogs. Paralogs are two genes that look very similar within the same organism. So this is the same organism. They have similar structures, they have similar functions, but they're in the same organism. That's a paralog. An ortholog is the same structure, the same sequence, basically, but different organisms. So orthologs, different organisms. O and O, organisms, right? Different organisms. Okay, so one of the things that I want to do with y'all, everybody's got their device with them, right? Because I think that's the end, right? That's the end. Oh, yeah. That's the end. Okay, so one of the things I want to do with y'all is I want you to go to Moodle. I can move this. Okay, so okay, here we go. So right here, under chapter 1, so open material for exam 1, under chapter 1, the very last thing says chapter 1, cell biology is the biochemistry of the cell. And it really is. I have my PhD in cell biology, but like being a biochemist, you really understand it. So what I want you to do is I want you to use your textbook and other reliable sources. Y'all know what reliable sources are, right? Not Wikipedia. To answer the questions that are on the slides. Now, how do you know which slide to answer? Well, I put you into groups, and you can look on yours. And then I also have the groups. I also put the groups. Where did I put the groups? Oh, here, right under seating charts. The groups are here too, if you don't want to like really focus in. But remember, I showed you this on like the first day. I showed you your groups. Okay. So you're going to look at the different slides. And what I want you to do is put a new text box. Everybody puts their own new text box and put your answer and then put your like first name, just so you know whose stuff is what. You don't have to get up. You don't have to move. You're going to work together virtually. Everybody in the class is going to contribute to this. It's like a study guide, right? And at the end of the day, I'm going to look over everything. I'm going to give you little notes and stuff. This is good, add that, whatever it is. And then everybody in the class has the same study guide. And it took you 5 minutes to do. And you have this study guide for the cell biology of the cell. Okay, sounds good? All right, so go for it. So if you know, to know which one you're going to do, you look, you're in a hydrogen group. So you'll look at the bottom corner and you'll see that your element is here, right? So that's a slide that you can work on. So there are like 20 different slides, right? So there are multiple slides. Once you're done with your slide, go and look at the slide above and go and look at the slide below and see if you can add anything to what the other groups were working on. So you do your slides and then look one above, one below to help the other groups, okay? Cool. Then we all end up with a beautiful study guide. Questions? Need help? Yes, ma'am. You can, all of them. So everybody's going to probably pick the ones at the top. So go down and look. There are more slides that have your group so that you can work on multiple slides. And then you go back to the other one, go look, see if you can add something to it. Yeah. Right, exactly. That's why I did multiple slides for every group. Yes. So this should be a good review of the cell biology that you need for biochemistry. And there's, oh, and I also put, I forgot to say this, I put a little link if you want to watch. It's a really nice animation of cell biology structure from start to finish. So if you want to watch that, but you don't have to. So it's a cool resource. And usually at this point I get to walk around, but I can't. It's really hard. I guess I can end the Zoom because that's what we're going to work on. And so class is done.